Reproductive biology is a fascinating field that delves into the intricate processes of creating new life. Did you know that the human body is composed of trillions of cells, each containing a unique set of genetic information? This genetic information is crucial for the development and functioning of an organism. In fact, the human body has approximately 37.2 trillion cells, each playing a specific role in maintaining our overall health and well-being. When it comes to reproduction, the fusion of male and female gametes is a crucial step in creating genetically diverse offspring. This process ensures that each individual is a unique combination of genetic material from both parents. In the animal kingdom, there are various reproductive strategies, ranging from external fertilization in aquatic species to internal fertilization in mammals. Interestingly, some organisms have the ability to reproduce asexually, allowing them to create genetically identical offspring without the need for a mate. This form of reproduction is advantageous in certain environments where finding a mate may be challenging. As scientists continue to unravel the mysteries of reproductive biology, new technologies such as cloning have emerged. Cloning involves creating genetically identical copies of an organism, offering potential benefits in fields such as agriculture and medicine. The study of reproductive biology not only sheds light on the mechanisms of life, but also raises important ethical considerations surrounding the manipulation of genetic material. By exploring the secrets of reproductive biology, we gain a deeper understanding of the fundamental processes that drive life on Earth. Sexual reproduction is a fundamental process that drives the diversity and evolution of life on Earth. One interesting fact is that sexual reproduction is prevalent among eukaryotic organisms, including plants, animals, and fungi, contributing to the vast array of species we see today. In fact, over 99% of all known animal species reproduce sexually, highlighting the significance of this reproductive strategy in nature. The fusion of male and female gametes during sexual reproduction results in genetic recombination, leading to offspring with unique combinations of genetic material. This genetic diversity is essential for the adaptation and survival of species in changing environments. Interestingly, the process of sexual reproduction is not limited to just two sexes in all organisms. Some species exhibit complex mating systems with multiple sexes, such as the mushroom Schizophilum commune, which has over 23,000 different mating types. This diversity in mating systems showcases the complexity and versatility of sexual reproduction in nature. In terms of numbers, the human body produces millions of sperm cells in a single ejaculation, each carrying a unique set of genetic information. On the female side, a woman is born with approximately 1 to 2 million eggs, but only about 300 to 400 of these will mature and be released during her reproductive years. These numbers highlight the incredible investment and complexity involved in the process of sexual reproduction. From an evolutionary perspective, sexual reproduction offers several advantages over asexual reproduction. One key benefit is the generation of genetic diversity, which allows populations to adapt to changing environmental conditions and resist diseases more effectively. This genetic variability is crucial for the long-term survival and evolution of species. In addition, sexual reproduction helps in the elimination of harmful mutations through genetic recombination and the shuffling of genetic material between individuals. This process, known as genetic purging, helps maintain the overall health and fitness of populations. Interestingly, some species have evolved unique reproductive strategies to enhance their reproductive success. For example, certain plants have developed mechanisms to prevent self-fertilization, promoting outcrossing and genetic diversity. This strategy reduces the likelihood of inbreeding and increases the chances of producing offspring with favorable traits. Overall, sexual reproduction plays a vital role in shaping the genetic diversity and evolutionary trajectories of species. By understanding the importance of sexual reproduction, we gain insights into the mechanisms that drive the dynamic and ever-changing world of life on Earth. Sexual reproduction involves the fusion of male and female gametes to create genetically diverse offspring. In humans, the process of fertilization occurs when a sperm cell penetrates the egg, resulting in the formation of a zygote. Interestingly, the sperm cell is one of the smallest cells in the human body, measuring about 0.002 inches in length, while the egg cell is one of the largest cells visible to the naked eye. The journey of a sperm cell to reach the egg is a remarkable feat, as it navigates through the female reproductive tract, overcoming various obstacles to reach its destination. In fact, only a small fraction of the millions of sperm cells released during ejaculation will reach the egg, highlighting the competitive nature of fertilization. Once fertilization occurs, the zygote undergoes a series of cell divisions to form a blastocyst, which implants into the uterine wall for further development. The process of meiosis, which involves the division of genetic material to produce haploid gametes, is crucial for sexual reproduction. During meiosis, homologous chromosomes pair up and exchange genetic material through a process called crossing over, leading to genetic diversity in offspring. In humans, meiosis occurs in the ovaries and testes, resulting in the production of eggs and sperm with half the number of chromosomes. The genetic diversity generated through meiosis is essential for the adaptation and evolution of species. In some species, such as plants, animals, and fungi, sexual reproduction involves the formation of specialized structures for gamete production. For example, in flowering plants, the male gametes are contained within pollen grains produced by the anthers, while the female gametes are located in the ovules within the ovary. The transfer of pollen from the male reproductive organ to the female reproductive organ is facilitated by various agents, including wind, insects, and animals. This process, known as pollination, is essential for successful fertilization and seed production. The timing of reproductive events is crucial for the success of sexual reproduction. In many species, environmental cues such as temperature, light, and moisture play a significant role in triggering reproductive processes. For example, some plants require specific day lengths to initiate flowering, while certain animals exhibit seasonal breeding patterns in response to changes in daylight and temperature. The synchronization of reproductive events ensures that male and female gametes are available for fertilization, maximizing the chances of successful reproduction. Overall, the mechanisms of sexual reproduction are complex and diverse, reflecting the evolutionary adaptations that have shaped the reproductive strategies of organisms across the tree of life. One of the significant challenges in reproductive biology is the issue of infertility, which affects approximately 10 to 15% of couples worldwide. Infertility can be caused by various factors, including hormonal imbalances, genetic disorders, and lifestyle choices. In vitro fertilization, IVF, has emerged as a groundbreaking technology to help couples overcome infertility, with over 8 million babies born worldwide through IVF since the first successful procedure in 1978. Another challenge in reproductive biology is the decline in sperm quality observed in men worldwide. Studies have shown a significant decrease in sperm count and motility over the past few decades, raising concerns about male fertility. Environmental factors such as exposure to chemicals, pesticides, and pollutants have been linked to this decline in sperm quality. Additionally, the rise in sexually transmitted infections, STIs, poses a threat to reproductive health, with over 1 million new cases of STIs reported daily worldwide. STIs can lead to infertility, pregnancy complications, and other reproductive health issues if left untreated. The issue of reproductive aging is another challenge faced by women, with a decline in fertility and an increased risk of pregnancy complications as they age. Women are born with a finite number of eggs that decrease in quantity and quality over time, leading to difficulties in conceiving naturally. Advances in reproductive technologies such as egg freezing offer women the option to preserve their fertility for future family planning. Overall, these challenges highlight the complex interplay of biological, environmental, and lifestyle factors that impact reproductive health and fertility in both men and women. Asexual reproduction is a fascinating reproductive strategy observed in various organisms, allowing for the creation of genetically identical offspring without the need for a mate. One intriguing example of asexual reproduction is found in the starfish, which has the remarkable ability to regenerate lost limbs and even grow into a new individual from a single severed arm. This process, known as fragmentation, enables starfish to reproduce asexually and regenerate damaged body parts. Another interesting form of asexual reproduction is budding, commonly seen in organisms such as yeast and hydra. During budding, a small outgrowth or bud forms on the parent organism and eventually detaches to become a new individual. The ability of these organisms to reproduce without the genetic contribution of another individual showcases the diversity of reproductive strategies in nature. In the microbial world, bacteria reproduce asexually through binary fission, a process where a single cell divides into two identical daughter cells. This rapid mode of reproduction allows bacteria to colonize new environments quickly and adapt to changing conditions. Interestingly, some plants, such as strawberries and spider plants, reproduce asexually through runners or stolons, which are horizontal stems that produce new plantlets. This method of reproduction enables these plants to spread and colonize new areas efficiently. In the animal kingdom, certain species, like the freshwater polyp hydra, exhibit remarkable regenerative abilities and can reproduce asexually through a process called budding. The hydra can continuously produce buds that develop into new individuals, allowing for rapid population growth. These diverse examples of asexual reproduction highlight the versatility and adaptability of organisms in perpetuating their species without the need for a mate. Cloning technology has made significant advancements in recent years, revolutionizing various fields such as agriculture, medicine, and conservation. One of the most famous cloned animals is Dolly the sheep, who was the first mammal to be cloned from an adult somatic cell using the technique of somatic cell nuclear transfer, SCNT. Dolly was born on July 5, 1996, at the Roslin Institute in Scotland and captured the world's attention as a symbol of scientific achievement. Since Dolly's birth, numerous other animals have been successfully cloned, including mice, cows, and even monkeys. The field of cloning has expanded beyond animals to include plants, with researchers successfully cloning various plant species to preserve rare and endangered plants. In agriculture, cloning technology has been used to propagate high-yielding crop varieties and disease-resistant plants, offering solutions to food security challenges. The ability to clone plants with desirable traits has revolutionized crop breeding programs and accelerated the development of new agricultural products. In the medical field, cloning technology holds promise for regenerative medicine and personalized therapies. Scientists are exploring the potential of therapeutic cloning to create patient-specific stem cells for treating a range of diseases, including Parkinson's, diabetes, and spinal cord injuries. The use of induced pluripotent stem cells, iPSCs, generated through reprogramming adult cells, has opened up new avenues for studying disease mechanisms and developing novel treatments. The field of cloning has also made strides in conservation efforts, with researchers using cloning technology to preserve endangered species and restore populations facing extinction. The Frozen Zoo, located at the San Diego Zoo Institute for Conservation Research, houses genetic material from over 10,000 individual animals, providing a valuable resource for future cloning and conservation initiatives. The development of cloning technology has not been without challenges and ethical considerations. Despite the scientific advancements, cloning remains a complex and inefficient process, with low success rates and high rates of developmental abnormalities in cloned animals. Ethical debates surrounding cloning focus on issues such as animal welfare, genetic diversity, and the potential misuse of cloning technology. As researchers continue to refine cloning techniques and address ethical concerns, the field of cloning holds immense promise for advancing scientific knowledge and benefiting society in diverse ways. Cloning technology has sparked intense ethical debates surrounding the manipulation of genetic material and the implications for individuals and society. One of the key ethical considerations in cloning is the welfare of cloned animals, as the cloning process can result in high rates of developmental abnormalities and health issues. Studies have shown that cloned animals, such as Dolly the sheep, often experience health problems and premature aging compared to naturally conceived animals. In fact, research has indicated that cloned animals may have shorter lifespans and increased susceptibility to diseases, raising concerns about the well-being of these animals. Another ethical concern in cloning is the potential loss of genetic diversity within populations. Cloning involves creating genetically identical copies of an individual, which can reduce the overall genetic variability of a species. This loss of genetic diversity can make populations more vulnerable to diseases, environmental changes, and other threats. Interestingly, only a small percentage of cloning attempts are successful, with many cloned embryos failing to develop or resulting in abnormalities. For example, the success rate of somatic cell nuclear transfer, SCNT, in cloning mammals is typically less than 5%, highlighting the technical challenges and limitations of cloning technology. The ethical implications of cloning extend beyond animal welfare to broader societal issues, such as the implications for human cloning and the concept of genetic identity. The prospect of human cloning raises complex ethical questions about individual autonomy, identity, and the potential for exploitation. As society grapples with these ethical dilemmas, it is essential to consider the far-reaching implications of cloning technology on both present and future generations. Sexual reproduction and cloning are two distinct reproductive strategies with unique characteristics and implications. Sexual reproduction, as seen in most multicellular organisms, involves the fusion of male and female gametes to produce genetically diverse offspring. In contrast, cloning results in the creation of genetically identical copies of an individual. One interesting fact is that sexual reproduction promotes genetic diversity through the shuffling of genetic material during meiosis, leading to offspring with unique combinations of traits. On the other hand, cloning produces identical genetic copies, lacking the genetic variability seen in sexually reproduced offspring. In terms of numbers, the success rates of sexual reproduction and cloning differ significantly. While sexual reproduction involves the fusion of millions of sperm cells with a single egg, resulting in a relatively low success rate, cloning techniques often have low success rates as well, with only a small percentage of cloned embryos developing into viable offspring. The field of cloning has seen notable successes, such as the cloning of Dolly the sheep, the first mammal cloned from an adult somatic cell. Dolly's birth in 1996 marked a significant milestone in cloning technology. In contrast, sexual reproduction has been the primary mode of reproduction for the vast majority of species on Earth, with its evolutionary advantages of genetic diversity and adaptability. Ethically, both sexual reproduction and cloning raise important considerations. While sexual reproduction aligns with natural processes and genetic diversity, cloning raises concerns about animal welfare, genetic uniformity, and the potential loss of genetic variability within populations. As scientists continue to explore the complexities of sexual reproduction and cloning, it is essential to consider the ethical, biological, and societal implications of these reproductive strategies. The future of reproductive biology holds exciting prospects, with advancements in technologies such as CRISPR-Cas9 offering new possibilities for genetic manipulation. CRISPR-Cas9, a revolutionary gene editing tool, allows scientists to precisely modify DNA sequences, opening up avenues for correcting genetic disorders and enhancing crop traits. In the field of assisted reproductive technologies, the use of artificial intelligence and machine learning algorithms is transforming fertility treatments by predicting optimal embryo selection for in vitro fertilization. Additionally, the emerging field of epigenetics, which studies heritable changes in gene expression without altering the underlying DNA sequence, offers insights into how environmental factors influence reproductive outcomes. With over 200,000 babies born through assisted reproductive technologies each year worldwide, the future of reproductive biology is poised for groundbreaking discoveries and innovations. In the ever-evolving landscape of reproductive biology, fascinating details and facts continue to emerge. Did you know that over 8 million babies have been born worldwide through in vitro fertilization, IVF, since its inception? Additionally, the success rate of somatic cell nuclear transfer, ESC, in cloning mammals is typically less than 5%, highlighting the technical challenges of cloning technology. With the development of CRISPR-Cas9, scientists now have a powerful tool for precise genetic editing, revolutionizing the field of reproductive biology. The use of artificial intelligence in predicting optimal embryo selection for fertility treatments is transforming assisted reproductive technologies. As we delve deeper into the realms of epigenetics and genetic manipulation, the future of reproductive biology holds immense promise for advancing scientific knowledge and improving human health.